So thank you, Andrea, and all the organizers. It's actually my second time participating in this, so I'm going to try and give a bit of an update. There's a lot to cover, but what I'm going to tell you about is actually under review. There are two different manuscripts, and I'll try and hit what I think are the high points. Let's see, it's not moving. Okay, so this work I'm going to tell you about is actually collaboration between quite a number of folks. I'm highlighting a few of them here, sort of in order of appearance. Gorka Lasso was a postdoc in my lab and shared with Barry Honig. Gorka has an independent position at Albert Einstein. Nick Tatnetti, of course, is here at Columbia. John Lambris is at UPenn, and Sandra Connolly is a clinician at CUMC. And this work stems from a general interest in my lab in understanding pathogen-host interactions, and we're interested. We think a lot about pathogens and how they manipulate cells that they infect for various reasons, experimental and practical, and obviously public health reasons. We focus our efforts on viruses, but we take a sort of a combination of computational and experimental approaches to understand the regulatory machinery and networks that control the interactions between viruses and hosts. And so on the one hand, we're interested in how viruses manipulate the host. On the other side, we're interested in how cells make decisions, and in particular, how cells of the innate immune system decide what genes to turn on and off. And so what I'm going to tell you about is work that is purely computational, obviously is very much within sort of the overall interest in virus-host interactions. The project sort of started, maybe 6 or 7 years ago, in a very close collaboration with Barry Honig. Barry's lab developed an algorithm called PREPI, stands for predicting protein-protein interactions. What PREPI does is it takes protein structure and gene expression and various annotations to predict whether pairs of proteins interact. And they applied it to yeast and human. And what Barry and I did as part of the collaboration is developed a new algorithm called P-HISPTer that allows us to predict interactions between viruses, pathogens and hosts. We applied it to all human viruses, and that paper is published and people can go and look at it. So viruses can manipulate cells not just through protein-protein interactions, but through mimicry. And mimicry is essentially the encoding of proteins by viruses that are structurally similar to the host, but are sequence divergent. And there are many examples of mimicry in the literature. Most of them are attributed to relatively large DNA viruses that have picked up host genes through horizontal gene transfer. And then those sequences have diverged. Given how central mimicry is, in particular for virus biology, we decided to take a sort of a comprehensive and computational approach to scan for viral mimics across Earth's virome. So there are about 8,000 viruses that have been cataloged, and we essentially mapped structure mimics across all viruses and their hosts. Just to give you an example of what we're talking about, when we look at phages, their proteins look particularly similar to E. coli, their natural host, but their proteins are quite relatively dissimilar to the structures used by yeast and humans. And so what we learn, beyond identifying I think something like 6 million pairwise structure mimics that are encoded by viruses, we identify sort of a general principle that viruses use a structure space that is, at least in part, defined by the host or their host range. So human viruses use structure space of human proteome, plants use structure space of plants, bacterial viruses use bacterial sort of structure space, and arboviruses, viruses that infect humans but are transmitted by insects, look like both insects and human. So viruses have really encoded the structure mimics as part of the molecular sort of machinery that they use to manipulate cells that they infect. And one of the interesting things that came out of this was that, as I imagine all of you know, viruses are encoded by both, can be encoded by RNA or DNA, positive and negative sense. RNA viruses, given the nature of RNA being less stable, RNA viruses tend to be much smaller in genome size. DNA viruses tend to be much larger, meaning they have larger proteomes. RNA viruses appear to actually utilize structural mimicry far more than their DNA counterparts. And coronaviruses in particular are among those that use that even more so. And one of the interesting things that came out of this is the discovery of about 140 proteins, human proteins that are mimicked by coronaviruses. I talked about this in last time I spoke, but for those who missed it, a lot of things will jump out. So DDX58, this is RIG-I. This is the cellular cytosolic receptor for RNA. It is critical for detecting viral nucleic acids and initiating the pro-inflammatory program upon virus infection, including coronavirus. There is, people recognize TMPRSS1. Of course, TMPRSS2 is, to some extent, plays a role in viral entry. And the things that really popped out initially were coronavirus mimics of complement and coagulation cascade factors. So complement, you can think of as essentially similar to antibodies. They bind antigen, but complement does so, coats particles in an antigen non-specific fashion. And essentially what the role of complement is to both directly kill pathogens, and in particular bacteria and parasites, but also to opsonize, to coat these particles, including viruses, in order to essentially instruct cells to phagocytose, internalize pathogens, and digest. So this was particularly interesting because during the initial SARS-1 and MERS outbreaks, there were multiple reports of coagulation and complement sort of syndromes. But one of the hypotheses that came out of these observations was that perhaps coronavirus encoded mimics may participate or contribute to some of the pathology associated with SARS-CoV-2. So we took a sort of a data and candidate-driven approach to discover disease determinants associated with the COVID pandemic. This is in collaboration with Nick Tetnetti. As I mentioned, this paper's under review, and people could read it. It's actually just went up on medRxiv early this morning. So what we do here, what I'm going to show you is data where we essentially take the predictions that we make on based on structural mimicry encoded by coronavirus, and leverage EHR data from NYP. I'm going to show you data from Colombia in particular. I can tell you that we're repeating the analysis across other hospitals, and it's consistent. So one of the reasons that complement was particularly interesting is that among human diseases that are associated with immune dysfunction, complement ranks among the top. One of the most prevalent complement-mediated diseases is macular degeneration. And this is both congenital and acquired complement dysfunctions. Among GWAS studies, complement genes repeatedly come up as risk factors for development of macular degeneration. And so it provides a relatively good sort of basis for studying whether history of macular degeneration provides a increased risk for adverse SARS-CoV-2 infection outcomes. Now, the other reason this is interesting is because macular degeneration is relatively common in the population, and given the large number of patients that have come through, the NYP system, Colombia alone has, I think we looked at close to 12,000 patients. So about 1 in 1,000 people suffer from macular degeneration, and so we have a large enough patient cohort we can actually do valuable statistics. So, okay, here you'll see this is cough. This is simply a patient that presents with cough and their risk of intubation. Of course, increased age, being male versus female, history of diabetes and obesity. These are things that have been reported as risk factors for essentially increased risk for both intubation and mortality, and you see that we essentially rediscover those things as well. This was essentially a sanity check to make sure that we are able to rediscover the same observations that were made in other cohorts. The part that's critical here is that you'll see that a diagnosis of macular degeneration results in extremely increased risk for both intubation and mortality associated with SARS-CoV-2 infection. Roughly 25 to 30% of people with macular disorders actually succumb to SARS infection. Coagulation as well is associated with increased risk. But what is, I think, quite compelling is that while macular degeneration, which as I mentioned is associated with enhanced complement activation, so it's a hypercomplement phenotype, complement deficiencies, which are extremely rare but yet happen, are associated with reduced risk. Now, I can't give you any p-value here. There's far too few patients for us to do a proper analysis, but of the 4 that tested positive, none of them were either intubated or died of SARS-CoV-2 infection. And remember, these are people who are thought of as being severely immunocompromised. Okay, so one way people like to visualize this type of data is by looking at sort of the rates of intubation. You see that macula is up there, and when we correct for both age and sex, of course, both of which have been attributed to the higher sort of risk to both intubation and mortality, and have been
Sagi, a 2-minute warning.
At least in part used to explain differences in susceptibility. They don't account for these observations. So one of the things we of course would like to do is we'd like to identify genetic markers of susceptibility to SARS-CoV-2 infection. As I mentioned, macular degeneration can be both acquired and congenital. UK Biobank recently released data on close to 1,000 patients, and so we did a sort of hypothesis-driven analysis looking for SNPs associated with complement and coagulation genes, and identified several, I believe we have a total of 10 here, that are significantly associated with adverse outcome. Two in particular are EQTLs for CD55. CD55 is a negative regulator of complement activation, and you can see that the EQTLs are actually associated with reduced expression. So I think one of the satisfying things here is that, biology, studying how viruses manipulate the host, can inform us about potential underlying conditions that are associated with risk factors. Of course, I haven't shown you in any way that mimicry is what leads to this association, but it at least drove the discovery of this. And to my knowledge, this is the first example of any genetic association with adverse outcome to SARS. I just want to finish off by one last thing. I was really quite fortunate to come in contact with John Lambris, who's at UPenn, actually where I did my graduate school, and he recently published sort of the first report of a patient being treated with a specific C3 inhibitor. This is a patient that was in his 70s with history of many comorbidities. Within 48 hours of treatment, the patient almost did a 180 and is now happily recovering. So we're now entering a phase 2 clinical trial and hoping to start recruiting patients at Columbia very soon. So I'll finish with that. Hopefully I've covered most of the folks that have helped with the work, and I'm happy to take some questions.
That's great. I think there are 2 questions already. Carol, Troy.
I'll stop sharing.
Okay. I'm on microphone. So on the coagulations, you had both hypercoagulable and people that don't clot. Are they at equal risk?
Right, that's a good question. So people who don't clot are similarly rare to folks who have complement deficiencies. So again, we have, it's more difficult for us to do a proper analysis on them. So
von Willebrand's is fairly common.
I'm sorry?
Von Willebrand's is fairly common.
Yeah, and so surprisingly, the numbers that have come through Columbia have been quite small. We don't have a good explanation for why that is, but that's very much something we're interested in looking at. And part of the effort to sort of broaden the analysis across other hospitals is to try and capture some of these folks.
So I don't know if you've talked with David Duguid. He deals a lot with coagulopathies. He's in our hematology.
I've not, but
he's great.
Very much interested in chatting with these people. Very, very much.
Thank you.
Thank you.
Kais Alaquari.
40% of the patients in the ICU are developing kidney failure, and a kind of kidney failure that is associated with proteinuria, which is like a glomerular disease. And complement is heavily involved in actually causing glomerular diseases. So I wonder whether you guys have looked at the association of kidney disease with what you had done.
Yeah, I'm glad you brought that up. So something I didn't mention is that the the reported clinical histories that have been associated with adverse clinical outcome with SARS include obesity, diabetes, history of kidney failure, history of heart disease, and many of these, including diabetes and obesity, are associated with acquired complement dysfunction. And we believe that part of the reason, at least part of the reason that there is this increased risk is due to the complement dysfunction in these patients. And which is one of the reasons why we think that this targeted therapy may be quite valuable for patients. Now, to answer more directly your question, whether we looked at patients with history of kidney disease, we didn't include in the paper, but yes, we have looked, and there's and we're not the first to do this, but there is a link. Appears to be a link between a history of a kidney dysfunction and increased risk with infection.
Andrea, it's David Ho.
Hi, David.
Sagi, thanks for the talk. And you must be aware of a paper from China describing the nucleic capsid protein of SARS-CoV-2 binding to MSP2, triggering the complement cascade and resulting in higher levels of C5a. I wonder if you have any comments about that.
I'm embarrassed to say that no, I'm not familiar with that paper, and so I'm not sure I can comment with too much clarity. But there are recently a number of reports that certainly complement is activated. Of course, IL-6 does reflect complement activation. So I don't think it's necessarily surprising, but
in the SARS and MERS literature, there's a lot of publications on activation of complement. But
sure.
This one, I should say, it's not a published paper. It was posted online, and you could find it. It reports nucleic capsid, a particular region of nucleic capsid binding to MSP2 in the lectin pathway. Mm-hmm. We have done some preliminary experiments to verify that indeed, in our hands, we see the same binding. Yeah. They have reported more data than that, but should take a look and yeah. I'd be interested in your opinion.
I mean, of course, I wouldn't be surprised. I should say I'm quite confident in the predictions that we make in the initial P-HISPTer paper. Our validation rate in terms of predicting protein-protein interactions was we had a true positive and true negative rate of close to 80%. We use a similar protein modeling pipeline to identify these mimics. So I'm quite confident. Of course, the structural mimicry does not mean functional mimicry, right? And in no way, I want to be really, really careful that in no way are we demonstrating anything about the virus necessarily impacting this. Having said that, we do identify mimics of mannose binding lectin. We do identify interactions with fibronectin, and of course, fibronectin is associated with both coagulation and complement activation pathways. So I'm not terribly surprised, but I'm also reassured that there are other groups that are observing similar things that are consistent with what we see sort of computationally and clinically. So.
Thank you, Sagi. Thanks to David. One last question from Margaret.
Hello, thank you so much for your talk. You've talked about genetic markers, acquired complement dysfunction, and you in particular mentioned data from the UK. Well, as you may know, the UK is having a similar problem to the US with regards to people of color suffering disparate death. You could also then look at Somali populations on the continent as well. So in terms of these acquired complement dysfunctions, I certainly see this in the US context where you have high rates of obesity, diabetes, and so on. But what about when you began to look at some of these other contexts?
Yeah, that's a really, really, really good question. Super important. If I didn't make it clear, there's a difference between congenital complement dysfunction versus acquired, and it's clear that the majority of the complement dysfunction are acquired and due to environmental, socioeconomic factors, and you know, when we look at the genetics, we don't account for any of that. Of course, there are genetic associations between, you know, that sort of predispose folks to some of these conditions, but you know, it makes it quite difficult to sort of tease those apart. Now, having said that, I think one of the difficulties. So if you've been following any of the GWAS studies, you'll know that none of them have been able to identify any genetic markers for association between any genetic marker and SARS-CoV-2 clinical outcome. And part of the reason is that, you know, 20% of people get hospitalized, about 50% of those get intubated, and of those, anywhere between 50 and 80% will die. And so it's very difficult to sort of identify any genetic component. And so by allowing the virus the biology to inform us on where to look, it allows us to identify these markers that, at least in part, contribute to those risk factors. I hope I answered your question. I know it's not particularly satisfying.